In that last part, I was going over how a complete, intact, original DNA molecule, represented by the two blue lines here, double helix. In terms of the replication process, you have origin of replication sites, multiple ones. They open up. Helicase opens up the double helix at the replication forks. Then the DNA polymerase molecules jump in and latch on to the single template strands here in blue. And one's the leading strand and one's the lagging strand. But the DNA polymerase molecule brings in the correct nucleotides, matching up the bases on the nucleotides with the bases on the parental strand nucleotides, because remember they bind complementary, A with T, T with A, G with C, C with G, and then you have the synthesis of the missing strand, which is termed a daughter strand here. These are replication bubbles. They spread and they spread out in both directions. When they reach the end of the DNA molecule, then you have 1, 2 intact daughter DNA molecules, both double-stranded, double helix molecules that will then go into their respective daughter cells, okay, for mitosis, DNA replication. So how an organism's genotype determines its phenotype. Back in our previous chapter or two, we talked about how genotype, the genetic makeup of an organism, determines the organism's phenotype, its physical or physical characteristics or traits. And of course, the expression of the genes and the genotype is influenced by the environment. So an organism's genotype, its genetic makeup, is heritable information because it's genes on chromosomes. Remember, different versions of the genes are called alleles. So that information is contained in the sequence of nucleotide bases in the DNA. And the phenotype, an organism's physical traits or characteristics, arises from the actions of the multiple proteins that are expressed from the genes or the alleles. That's why you have white or purple colored flowers on those peas, or say the tan brown coat on this mammal, because the genes are expressed to produce the physical proteins that have some characteristic or trait. So what you see is the phenotype determined by the genotype. Remember the alleles on Mendel's peas? So the overall idea is that the genetic information, the genes or alleles on the DNA, get converted into a secondary informational molecule, RNA, specifically messenger RNA, mRNA, that we'll talk about. And that gets converted into the physical protein or polypeptide that gets converted into a protein. And the protein has some physical trait or characteristic, like white color or purple color. So the overall process of the genotype becoming the phenotype, we're talking about the expression of genes or alleles that will become proteins. Polypeptides, remember, long chains of amino acids that get modified and become the functional protein. So DNA synthesizes proteins in two basic steps, or gene expression, if you will produce proteins in two basic steps or stages. One is transcription, the information on the gene or allele. Its information, sequence of bases, is transferred onto a copy of that genetic information, which is called RNA molecule. We'll soon call it an mRNA molecule, but it's just information. It's a series of bases. Okay? And that's transcription. It happens inside the nucleus of eukaryotic cells. And then the translation part, stage, is where that genetic information on the RNA or mRNA molecule gets converted into the physical product, the polypeptide that will become a protein. So we'll look at these two steps, transcription and translation. Okay? So this is the flow of genetic information. The function of a gene or allele is to dictate the production of a polypeptide that will become a protein. So the genetic information in DNA is transcribed onto the RNA molecule and then translated into the polypeptides, which become the functional protein, which is why organisms have their phenotypes, physical traits, characteristics, determined by expression of their genotype, the genes or alleles, through transcription and translation. Excuse me. So the language of DNA and RNA is the linear sequence of nucleotide bases. Here we zoom into one region of a gene, and we see that one strand of the gene, only one strand gets coded, by the way, to produce the mRNA molecule that will produce the polypeptide or the protein. We'll get into those details. But if we zoom in, this is an informational molecule, the DNA molecule, which means genes or alleles are just informational sections of the entire DNA molecule. So it's like a recipe to produce a product, a polypeptide or protein. And the recipe, the language, is the sequence of bases. Kind of like the letters of the English language are a language, or produce words that make sense. Okay? So the information in the DNA molecule or the genes is comprised of this unique sequence of bases. And remember, there's only 4 bases. But the DNA molecules, the DNA in your cells, have all the information to make you, which is a pretty complex large organism. So you need a lot of bases to hold a lot of information for something like us. And we have approximately 3 billion base pairs in our DNA molecules. So a typical gene or allele has thousands of nucleotides in a specific sequence that will code for a particular protein through transcription and translation. So the DNA nucleotides get transcribed, producing the RNA nucleotides on the RNA molecule that gets translated to produce the unique sequence of amino acids. If you remember polypeptides, protein synthesis, or polypeptide, or protein, is the polymer made up of the individual monomers amino acids. But proteins, polypeptides, are produced by reading the language of the nucleotides of DNA and then the language of the nucleotides of RNA and converting that into the unique sequence of amino acids for a particular polypeptide, which will become a protein. So we'll look at this process: transcription, then translation. So this is the flow of information from gene to protein based on triplet codes. So a triplet means 3s. Look at this diagram here. You have 3 nucleotides on the DNA molecule or the gene, 1, 2, 3. And one by one, they're converted into a grouping, a triplet of 3 nucleotides on the RNA molecule. So the process of transcription from DNA nucleotide to RNA nucleotide, that's a 1-to-1 relationship, as we'll see. But when the RNA nucleotides are converted into the amino acids, that's a 3. And what I was saying there at the last bit, I had to take a little pause. The grouping of 3 nucleotides on the RNA molecule, that grouping of 3 nucleotides is referred to as a triplet code or a triplet codon. So that triplet of bases on RNA codes for, hence the term codon, codes for a particular specific amino acid. So when we go to translation, RNA to amino acid, that is a 3-to-1 relationship. Okay. So instructions for the amino acid sequence of a polypeptide are written in DNA and RNA as 3 base words referred to as codons. And we really only use the term codon when we're talking about the RNA molecule. So codons in DNA are transcribed into complementary codons in the RNA molecule. And all that means is that every A base becomes a U base, not a T base here, because now we're talking about gene expression to produce a protein, not DNA replication. So every A base becomes a U base in RNA, but notice the C base still becomes a G, and the G base still becomes a C, and if there was a T base over here, it would become an A. But RNA has the 4 bases of U, G, C, and A, whereas the bases of DNA are A, C, G, and T. They just don't happen to have any T's in this little sequence here. Okay, so the RNA codons are translated into the specific amino acids of a polypeptide, as we'll see. And how is this done? It's done through what's called the genetic code. So this is essentially the universal code that's used by essentially all organisms on Earth. And it's a set of rules that allows for the conversion of the nucleotide sequences, the 3 bases in RNA, those codons, into a specific amino acid and an amino acid sequence as the polypeptide chain is being built. So there are 64 triplet codons, and those are indicated here like the UUU and the UCU, etc. 61 of the codons code for a specific amino acid. You don't need to know them by name, but just recognize the 3-letter abbreviations are different for the different amino acids, like PRO for proline, GLN for glutamine, etc. And there are 3 stop codons. UAA, UAG, and UGA are stop codons, so they will stop the synthesis of a polypeptide as the mRNA molecule is read, as we'll go into more detail in a bit. And then there's a start codon, UAG, which is always at the start of a particular sequence a sequence of codons in an mRNA molecule. Again, this should make sense as we proceed. But these triplets of bases on the RNA molecule, and you know it's the RNA molecule because of the U's, there's no T's, is what is called, or what are called, the codons, and they are the genetic code for essentially all living things on Earth. Okay? In other words, the expression of genes to produce proteins follows this same language of essentially all living things on Earth. There's some minor exceptions, but there are exceptions to this general rule. Okay, so the genetic code is shared by organisms from the simplest bacteria to the most complex plants and animals. The process is the same, essentially. So because the diverse organisms on life share this genetic code, it's possible to program one species to produce the protein from a completely different species by transplanting DNA, by genetically altering the genome of organisms. And they've done this in the lab quite a bit. Your textbook talks about glowing pigs, because a gene from a glowing jellyfish has been removed from the jellyfish, first of all, essentially, and inserted into the genome of the pig, and the pig then expresses that gene for a green glowing protein, so you get green glowing pigs. That in itself is more of a curiosity, but in terms of importance in medicine or agriculture and or in research, scientists can mix and match genes from various different species, which is a technology, biotechnology, that's really useful in genetic engineering. And actually that's how essentially all of our insulin is produced today. The insulin gene, the information is obtained from humans, and that gene is inserted into the genome of a bacterium, E. coli. They grow many, many bacteria rapidly and quickly in the lab. And they break apart the bacteria and extract the insulin, put it in a bottle, and sell it to us.